Hello guys, today we will be talking about promoters. So whenever we talk about transcription in prokaryotes or eukaryotes, promoters always come into our minds. So what is a promoter and what is its significance? In general, a promoter is a DNA sequence that starts the transcription. We can explain this in an easy way. But if we dive deep into the science of promoters or biology, we have two types of promoters to talk about, and they are always found together in the transcription. And so, number one is a core promoter, and the other is proximal promoter elements. Let's talk about the core promoter. Core promoter is the DNA sequence where RNAP2 or RNA polymerase II and transcription factors come and bind so that RNAP can start the transcription process. So you see, it's really important type which cannot be skipped because otherwise we won't have any transcription. So in eukaryotes we have Tata box, while in prokaryotes we have Pribnow box as the core promoters. Now, let's talk about proximal promoter elements. Proximal promoter elements are the type of promoter that enhance the transcription of a gene. The difference between two types of promoter is that PPE, or proximal promoter elements, bind to gene-specific transcription factors, such as we have a GC box, which binds to SP1 transcription factor, and CAT box, which binds to CTF transcription factor, while core promoters bind to general transcription factors such as TF2A, B, D, F, etc. Another query that you might have if you have already studied enhancers, it will be that what is the difference between proximal promoter elements and enhancers? I mean, they are both cis-regulatory sequences, so if they both enhance the transcription of a specific gene, then why do we even have two? We can be enough with just one of them, right? So now let's discuss this query. When we talk about proximal promoter elements, we always have to remember that they are position dependent and orientation independent. So even if we move it like 180 degrees, it is still gonna enhance the transcription of the specific gene, while enhancers are both position and orientation independent. So, for instance, I have a DNA sequence, I have a Tata box as a core promoter. The proximal promoter elements will always be found approximately 250 base pair upstream of the core promoter, while enhancers will be located even further upstream. So, for instance, if we take the proximal promoter element from the original site and put them even further away from the core promoter or transcription start site, they would not be able to enhance the transcription of the specific gene. While enhancers, no matter how far they are, they would still be able to enhance the function or the transcription of the gene because they are position independent. So this is the main difference in between two cis regulatory sequences. I hope you get this all and thanks for listening.